Hello, students. I thank you very much for your time and decision to be with me. If you have any biological topics that you need to study with me, put it on the comment area. In this lesson, we are going to study gene expression. Do you have an idea about gene expression? I think so. You have some ideas about gene expression from our previous class on transcription and translation processes as first and second steps of gene expression respectively. What is gene expression? Gene expression is the process in which genetic information from a gene is used in the synthesis of functional gene products. These functional gene products are proteins and functional RNA. Functional RNA includes small nuclear RNA, double-stranded RNA, messenger RNA, ribosomal RNA, and transfer RNA. What can regulate gene expression? Genes can be regulated by transcription factors and transcriptional interference. We know that all our body cells contain all genes, but all genes cannot get expressed in each of our specialized cells. Most scientists believed that all our body cells supposed to contain more than 35,000 genes. The genes whose product is required by that specialized cell can be switched on and expressed, but the other remaining genes whose gene products do not required by that cell must be switched off and remain dormant. For example, the gene that controls the color of our iris is present in all our body cells, but the gene is switched on and just gets expressed only the iris of our eyes. But the gene that controls the color of our iris must be switched off in our other body cells like skin cells, nerve cells, cardiac cells, muscle cells, alveoli, and etc. How are genes switched on? Very often, genes are switched on by transcription factors that are present in the cell. These transcription factors are usually proteins and that are designed to bind a regulatory sequence of DNA near to the gene they influence, what we call promoter sequence. Gene switching on process operate in the following way. First, RNA polymerase must bind to a promoter sequence of DNA near to the gene needed to be transcribed or expressed. Second, basal transcription factors must bind to a promoter sequence of DNA near to the gene to be activated. And thirdly, transcription factor activator must bind to the promoter sequences. When this all placed on promoter sequences, transcription factor activator activate basal transcription factor and hence basal transcription factor activate RNA polymerase. In this case, RNA polymerase moves away from DNA and transcription factor complex along the gene. Then the RNA polymerase transcribes antisense strand of the DNA as it moves along the gene. In this moment, the gene is being expressed. How are genes switched off? In this lesson, we are going to study only post-transcriptional interference. Besides transcription factors that promote the expression of genes, there are other factors that can act to repress gene action or that can act to switch off genes. One group of substances that does this is known as short interfering RNA or siRNA. These RNA molecules are unusual because they are very short, only about 21 to 23 nucleotides long, and these are double-stranded molecules. These molecules do not act on the gene itself, but it interfere with or silence mRNA once it has been transcribed from the DNA, what we call post-transcriptional interference. You know, if mRNA is prevented from translating its codons into amino acid sequences, then the protein for which the gene codes cannot be built. This means the gene has effectively been silenced, what we said before, post-transcriptional interference. The RNA interfering mechanism is an important biological mechanism in the regulation of gene expression. Biologists think that the action of short interfering RNA takes place as the following. First, double-stranded RNA is produced in the nucleus from a range of genes. It is then split into the very short lengths that characterize short interfering RNA by an enzyme called dicer. Then the antisense strand of the short interfering RNA then binds with a complex of molecules called RISC or RNA induced silencing complex. The short interfering RNA binds with mRNA and allows RISC to degrade or cleave mRNA into smaller fragments. As we can see from the diagram, double-stranded RNA binds to the protein dicer and gets cleaved into smaller fragments, what we call short interfering RNA. One of the RNA strands, that is, antisense strands of the RNA, is loaded into RNA-induced silencing complex or RISC. Then RISC links antisense strands of short interfering RNA with mRNA strands by base pairing. Lastly, as we can see, mRNA is cleaved and destroyed. This results in no protein synthesis at all. From RNA interfering mechanism, most biologists believed that short interfering RNA hold a great deal of promise to treat AIDS and some cancers. Researchers have already shown this action. They can use short interfering RNA to prevent replication of HIV in cultures by silencing some of the genes on the HIV virus, or to prevent some of the human genes on which HIV virus depends on. They can also use short interfering RNA to silence genes associated with cancer. That means to silence oncogenes. So, silencing oncogenes results in many new possible treatments for many cancers.